Our next speaker is Dr. Bradley Strauss, who became familiar to many of you yesterday at the award ceremony. And he will be discussing completing the task, Canadian style, from cardiac researcher to Medtech startup. Dr. Strauss?
Thank you very much. First of all, it's a great privilege to be here, and I'm, again, very grateful for the very generous award that was given to me yesterday. As a member of both the American College of Cardiology and the Israeli Heart Society, as well as the third Canadian Cardiovascular Society, I'm very glad to present here in this very distinguished session. I'm going to talk a little bit about a personal journey that I have. It'll be different than the other talks that we've had. And at the end of it, I'm going to try to bring together of how I think this will work out somewhat in the future. And it's particularly geared towards fellows who are in the audience. And as you know, I have great interest in fellows, and I think that this is a very interesting way to engage the fellows. So these are my disclosures. I have a startup company, and I have IP in the company. So my journey begins, if you can, oh, there it goes, about 14 years ago. I pray this works. This is the key of the whole day. Can you see if you can get that to work? Thanks. I would ignore it if I could, but it is important to this, so let's see. Are you going to do it by hand here?
You can write it outside the PowerPoint.
I think it's embedded in it, so. Do you give up there at the back? Okay. So, okay. Are you going to try, or you can't do it? Okay. So what I was about to show, if you would have seen it, was something that occurred about 14 years ago. I was working in my lab trying to think of new ways of how to open up chronic total occlusions. And I came upon this idea of, what if I injected collagenase directly into the artery where the blockage is? Would it be able to sufficiently soften the occlusion and then allow me to do the procedure with a softer guide wire? I worked on it for a while. I looked at it for 10 minutes after I'd given it a try. It didn't work after a long series of attempts. And then one day I waited for 3 days because I thought maybe it needs a little bit of time to work. And it was around the time that actually Dr. Segev was working with me. And to my great delight and surprise, I was able to cross the artery with a very soft guide wire in a very short period of time. I don't know, is this coming up here? You going to try again? Oh, okay. Okay, it's okay. We'll move forward, don't worry. I'll do that. So I was successful, and I was so happy and so thrilled when I actually saw something that I had thought about actually work and practice in the research setting. But then I thought, what next? What do I do next to move this therapy along? And it led me down this entire road of what is innovation, how does it happen? And I apologize if I seem presumptuous talking about innovation here in Israel, which is really the startup nation itself. But I have learned a lot here, and I've learned a lot along this road, and I think that we can share that together. So we have the next slide. I'll do it. Okay, so this leads me to the area of what's called translational research. There are a large number of definitions of it, but this is one that I liked recently that came out of an internet search I did. It turns early-stage innovations into new health products and advances the development of an innovation to the point where it may become attractive to others, either venture capital firms, industry, or public-private partnerships, to take up the challenge to produce it for the market. And this is from the Wellcome Trust website. This is quite a unique definition of translational research. And I think it really reflected what I'm going to try and talk about here, what my thought about translational research is, and how it's not just enough to get the idea, but you have to move the idea along. So my greatest frustration for many years was that my inability to open chronic total occlusions, which are coronary arteries that have been blocked for 12 weeks at least. And you can see here an arrow pointing to the chronic total occlusion. We know that PCI success rates in this subset of lesions are relatively low, 50 to 70%, and the guide wires are unable to do it because they cannot puncture through the very hard plaque that blocks it. In addition to it, the strategies that we have adapted are to take very stiff guide wires, which can easily perforate through the artery and cause a lot of difficulties. And even when you're successful, you actually use very long procedure and fluoroscopy times as well as heavy contrast. I think we're going to take till like 2 o'clock till we get this done, though. Nah, you know what? It's okay. Hopefully the rest will work. It's okay. Thanks. So the idea was, what is actually blocking the artery? And from a number of pathology studies, both experimental and human pathology studies, I realized that collagen was really the main constituent that was blocking the progress of the guide wires. And with that in mind, I hope that this idea of bacterial collagenase, which we call this product MZ004, would be useful to soften the occlusions and allow us to be successful in crossing them. And this enzyme collagenase comes from a bacterium known as Clostridium histolyticum, and it's an enzyme that has 2 different isoforms, and as well as a small amount of additional proteases that are produced. And the enzyme works by actually degrading collagen within the chronic total occlusion, allowing you to soften the occlusion enough that you will soften it within the occlusion but limit the damage to the outer arterial wall. And based on that, I went through a series of steps with a lot of interactions with others to come up with what I think the steps are of Innovation 101. The first is, and you'll see this be a constant theme here, you need financing very early on in order to do it. Secondly, you have to move it forward. You really have to assess, is there a clinical need? Is there a market size that will make investors interested in it? Is it patentable? Is there a preclinical model that you can develop the idea? Then you need more financing. Then you have to decide, how do you do the manufacturing? What about the regulatory status? First-in-man trials, more financing because it costs a lot more at that point, phase 2 trial, yet more financing, and finally, hopefully, the pivotal phase 3 trials. These were steps that I never learned in medical school. I never learned it during my cardiology residency or fellowship, and it's been on-the-job training. So the important steps of innovation, I would say, first of all, you have to start out with the idea. I think everyone in this room has very excellent ideas that could be pursued. The idea has to then be translated into preclinical studies and first-in-man studies. And finally, it has to be operationalized. In order to do that, you have to build a team. This is something also we're not used to, working with people in other disciplines like lawyers, regulatory people, manufacturers, business people, and fundraisers. Then you have to actually execute the idea, and that's arranging phase 2, phase 3 trials, which for someone who does basic research is also a new experience. And finally, what's the exit going to be? So what really makes a good idea? How, if you have an outstanding idea, should you pursue it in the cardiology realm or any realm? First of all, it has to address an important clinical problem, and that's based on clinical experience and market analyses. Second of all, it should have a solid basis in the pathophysiology of the problem, so you have to understand the basic and clinical literature in the area. Thirdly, it has to be able to be tested prior to patient trials in a preclinical model, and often that involves the collaboration of engineers as well as modelists. And then it has to be patentable, and the main issues tend to be obviousness and prior art. And it has to have a clear regulatory pathway and strategy, and you have to be familiar with the regulatory pathways in several different areas of the world, including US, the Far East, and Europe. Finally, you have to be able to use GMP manufacturing. It has to be done with reasonable scale-up. And you have to try investors and funds, which can come from a number of sources, including grants, etc., and then clinical trials, and finally, the exit strategy. So that's what you have to learn as an innovator, something that very few of us have learned, I think, prior to in our training or cardiology career. So I felt that the CTO revascularization strategy that I was working on had clinical experience to back it up, showing that if you improve CTOs, you improve quality of life, decrease need of bypass, improve LV function. The data on LV survival is not really there. And unfortunately, at this point, there's really been no randomized trials versus other forms of revascularization by surgery or compared to medical therapy. Now, is there enough there? The second question is there enough there worth looking at this problem? This was work done by Paul Pfeffer when he was a fellow in Toronto. Now he's at Teleshomer. We looked at this paper published in JACC a couple of years ago, looking at what is actually the prevalence of the problem. And it works out that it's about somewhere between 15 to 20%, depending on whether you're looking at it in Canada or in Sweden. So that's a fair number of angiograms that have these chronic total occlusions. Certainly something to pay attention to. And when you calculate in the U.S. that there's 3.6 million angiograms in 2010, that makes that, if you give our numbers, somewhere around 500,000 chronic total occlusions, which is quite a large number. Now, I would never say that all of those, or even the majority of those, need to have some therapy, but there certainly is a number of these cases that need some sort of attention. So currently, if we look at the way we treat it now, most of the patients, 2/3 of them are treated by medication, bypass surgery in 25%, and angioplasty, which is my area, is relatively modest at less than 9% in most of the sites. And overall, only about 30% of chronic total occlusions are revascularized, so I think that there is an opportunity to help a lot of patients who are otherwise not getting adequately treated for symptoms. When you calculate the market size, and this is something that you do in terms of with business people in order to try and attract investor money, which is important to move these things forward, the numbers are somewhat artificial, I think, but still show that for investors there is reason for them to support projects like this. So for 6 years, I engaged in preclinical studies. We published the first one in Circulation. Amit Segev was part of that, and the second article that he published in another journal and looking at different preparation. And we showed I think in a very good way that if you treated these rabbit arteries, which unfortunately I couldn't show you from the first slide, with collagenase versus placebo, you were able to significantly increase the crossing when you used collagenase without damaging deeper layers of the vessel wall. So we had a preclinical basis for what we were doing. At that point, then, we had to also engage in patents. And patents is a very expensive and very challenging area. These were a couple of the U.S. ones that we developed. And the costs, which were quite astronomical in my way of thinking of it from the beginning, ended up being over $200,000. So you definitely need support beyond your brother-in-law and yourself, which is how I started this. The regulatory is something that I also, as cardiologists, I don't think we're used to going to the FDA or to Health Canada. We looked many times you have visits there. You have to make your protocols and your plan very much in keeping with the strategy that will ultimately allow your product to be approved in various jurisdictions. And this is a very ongoing collaborative process. Now, the idea of where do you actually make something that you can put into patients, initially we just took stuff out of a bottle that we used for cell culture. And eventually I came upon a facility in Israel and Jerusalem that manufactured the GM collagenase, allowed us to do it in these human trials, which was a very challenging place to find anyone in the world that actually would make a grade that we could use in patients. So the first study was done, completed in 2011, was 28 patients, all of them had previously failed. And it was really a safety and efficacy study, but we showed that 21 of the 28 were actually able to successfully cross with this type of therapy in patients that had previously failed. Perhaps more importantly, it was done in 80% of the patients using very soft guide wires rather than these stiff spears that we use, and a very low level of adverse events. And this was work that I did with Uzi Osharov, who is in Ashkelon. We published this in Circulation about 3 years ago. According after this, where did I go? Well, I had to make a team. And these are the individuals that have been working for the last few years, regulatory, clinical trials, and a CEO. And we had to set up a place outside of, it's connected with the university, but an office that we call ourselves Matrzyme. And finally, I want to talk a little bit about financing, which is a huge issue and challenge for anyone doing this kind of work. I started this work in 2000, it's 15 years ago, with a $10,000 grant from St. Michael's Hospital in Toronto. Then it was financed through the preclinical work with about $156,000 Canadian by the Heart and Stroke Foundation. The first-in-man study had, now the money starts to go up, almost $300,000 to do the first-in-man, about 28 patients. Now we are in a phase 2 trial It's $9.5 million to do the manufacturing, regulatory, and phase 2 study. And ultimately, we will have to do a $15 million raise to do the phase 3 study. So it starts to add up from $10,000 where you start. And currently, we're in a phase 2 trial. This is a trial run in Canada in 10 clinical sites, as well as 2 clinical sites in Holland and hopefully 2 in Israel. Chris Buller is the head. It's a 90-patient, but 50 patients are randomized. The protocol is one that we developed in the animals. I'm not going to go through it now, but we're predominantly interested in safety as well as efficacy between the collagenase versus placebo. It's not the pivotal trial. It's really one step along the way. At this point, we've done about a third of the trial, including 5 of the 50 randomized patients. So I just want to show you for a moment the way we do it. I'll actually show here. This is chronic total occlusion. You can see in the mid-LAD, there is, it doesn't look like a very long or tough occlusion, but it actually took a host of very severe, tough guide wires and could not penetrate through. There was very, very hard lesion. This was a training case, so the patients, the doctors learning how to do it. So they do get 1 of 2 doses of collagenase. So this is not one of the placebo trial cases. This is the early training one. Then you can see now the. Okay. Let's see if it comes through here. No. Oh, let's see. Okay, so you can see the little dot there where the injection catheter is now inside the plaque of the chronic total occlusion. We inject there. Now there's no effect on the lesion from the injection. You can see everything looks very clear and the occlusion's untouched. Next day the patient comes back, and within about 2 minutes a soft guide wire is crossed, and in this case the final result is very pleasing to the eye, so I'll show it to you. And you can see that a very successful case that showed it, again reproducing what we initially showed in the animal work. So based on this, I've been very interested in how do you create a culture of innovation, how do you do it so that you can teach the lessons and get others engaged in what you want to do. It takes mentorship. I think it's very important to start teaching innovation at the medical schools and PhD programs, promote the clinician, investigator, innovator career stream, which we have, University of Toronto, dedicated innovation programs which are needed, which are modeled after the biodesign program at Stanford, now currently done in Jerusalem as well, and high-quality assistance for innovators, the thing of team building, which again is a very new concept for people who only do clinical work, as well as innovator-friendly IP policies. I think there are a whole host of great mentors around. John Simpson being a huge one, Paul Yock doing wonderful work in the States. Israel itself here has fantastic mentors. Moshe Flugelman has taught me a lot over the years, as well as Chaim Lotan, who does a meeting really dedicated totally to innovation. Gary Gashoni in the Bay Area has also done a lot of teaching and a great model to look with. And finally, Brian Courtney at my own institution, who is an incredible resource. Now, how is Canada performing? And I think this is very important from the point of view. We do great basic research in Canada, but we are a net importer of medical devices. This is looking at devices particularly. We have imports of $7 billion, whereas we export $2 billion. So there's a $5 billion difference between it that we for deficit. And smaller countries actually are doing much better in terms of exports. Ireland, $9.6 billion for a population of 6.4 million. And even in Israel, with a relatively modest number, about a quarter the size of Canada in terms of the population, has roughly the same number of exports as we have. The U.S., by the way, is $100 billion, employs 365,000 people, so this is a huge area and one that I think the Canadians really need to be more engaged in. And based on that, we've developed the Sunnybrook Medventions program. The key components of this are we have an Innovation and Commercialization Fellowship that we will be starting in 2016, and I hope that Israeli fellows will be part of this. We have clinical shadowing services for engineers, entrepreneurs, and researchers. We've developed a community educational network events where we have seminar series and a research day, and we have a whole series of digital medtech entrepreneurship community events in order to spread out the word of what we're doing. So this innovation series one, and I'm coming to the close of my one very quickly, Every month we have a entrepreneurship educational module where we bring people together to learn about how to do innovation. This is done with a number of industry speakers and speakers who are very experienced innovators. And it's a participant of a number of different companies and academic institutions. We're about to engage on a commercialization fellowship, an innovation fellowship, involving the business school at York University, University of Toronto, as well as engineering schools at the University of Toronto and York University. And this fellowship will involve 1 year of making teams of master's students in business, engineering, and fellows. And within that 4 months, they will learn about needs finding assessment, product design development, business and development, and the idea that we will be able to further the training of these individuals to identify the clinical need, invent disruptive technology, and to implement it. And, sorry. And this finally is the Schulich Innovation Research Day, which will be held on June 15th this year. This gives us an opportunity to highlight the work of many different experts in the area, and you can see that the Chaim Lutan from Jerusalem is attending. I just want to attribute the great contributions of my collaborators at Sunnybrook Hospital in Toronto who are working on it. And so in summary, I want to impress on anyone, I think the importance of the translation of novel research requires some innovation skills. These innovation skills need to be taught as a separate entity through multiple modalities, including focused multidiscipline fellowships, as well the value of these types of specialized innovation skill programs, such as Medventions, though I think will require careful evaluation to really determine in a prospective manner whether we are in fact succeeding at increasing innovation. The outcome of the CTO2 Phase 2 trial, we'll just see where that will be. I'll be able to report on that, I think, next year in the first quarter. Thank you again for the invitation, and thank you for your attention.
Thank you very much, Dr. Strauss. In view of the time, I think we're going to have to defer the questions. If people have questions, please approach Dr. Strauss if he has a moment. And in closing the session, I'd like to first plug the fact that we have 3 great sessions that are about to happen, sponsored lunches. I'm sure they're very healthy lunches, in halls D, E, and F on atrial fibrillation, heart failure, and lipid therapy. And my last comment is just, again, to reiterate our thanks for the opportunity to have this joint session. I think it was marvelous from an academic and practical point of view, and I'm hoping that we can continue this collaboration in the future. Thank you very much.